Hi, welcome to our presentation. We're here to talk to you about wine yeast, more specifically, how genetic modifications are changing the overall effectiveness and viability of differing yeast strains within the realms of winemaking. We'll be covering a wide array of subjects on the matter, such as details on how yeasts have been utilized within the winemaking process throughout the history, favorable traits and qualities in commercial yeast strains, how traditional selective breeding has been utilized within these strains, some current approaches to genetic engineering, and the overall future of wine yeasts and how they are changing. But firstly, some background. Let's have a chat about what the purpose of yeasts are in winemaking and how they work within the fermentation process. The role of yeast in winemaking is very simple: to convert the sugars present within the grapes into alcohol and carbon dioxide. Winemakers rely on yeast to create their desired style of wine just the way that they like it, with differing strains allowing for different characteristics to be imparted into the wine being created. Yeasts can come from different places too. Commercial yeasts see an abundance of use throughout New World winemaking in order to maintain control and fine-tune their product, whereas the utilization of wild yeast is also prevalent in order to obtain characteristics of which are reminiscent of the region of which the grape is grown. So, with that in mind, let's move on to some history.
Historically, wine was fermented with the yeast that naturally occurred in the vineyard or that was left over in the fermentation vessels, known today as a wild ferment. The first recorded winemaking traditions were over 8,000 years ago in Georgia. Here the wines would be fermented in earthenware vessels known as quavery. These quavery were buried once filled with grapes, where they would ferment for around 3 weeks. They wouldn't inoculate. The naturally occurring yeast would do that job. It wasn't until 1680 that yeasts were observed under a microscope. It took another 100 years, in 1789, for Antoine Lavoisier to describe the process of yeast fermentation as the process of converting sugar to carbon dioxide and ethanol. It took another 100 years again for Hermann Müller-Thurgau to introduce the process of inoculating wine with purebred yeast cultures in 1890.
Now, let's look into how artificial selection has played a role in the development of current yeast strains used around the world. Artificial selection has been taking place for thousands of years in order to promote desirable qualities in offspring for things such as livestock, crops, and domesticated animals. With the development of agricultural science in recent centuries, a greater understanding of these practices has allowed for the informed and deliberate selecting of desirable traits in yeast strains used today. Understanding the lineage of yeast strains used around the world has proven to be a bit more difficult than understanding that of livestock and food crops, as the selection of beneficial yeast characteristics has not been recorded to a great extent. A reason for this is the lack of understanding about microbial organisms before the 17th century. However, the development of scientific understanding between now and then has led us to a point where geneticists can now undertake genome sequence analysis and create phylogenetic trees that reveal how traits have evolved over time through selection. A lot of research has gone into mapping the lineage of the industry's most important yeast species, Saccharomyces cerevisiae. Such research includes work by Professor Kevin Verstrepen in Belgium. Verstrepen's work involved the genetic analysis of 157 Saccharomyces strains and found that these strains can be divided into 5 sublineages, all of which are genetically different from the wild strains and originate from only a small few yeast ancestors. This research suggested that artificial selection was key in bringing forward the yeasts that we see today. Ancient methods used in winemaking have given rise to the new yeast strains. Such methods include that of backslopping, which is where a successful and desirable naturally fermented wine would be used to inoculate a fresh juice or must, creating a nutrient-rich environment far removed from the yeast's natural niche, which was a perfect setting for the domestication of that yeast. This highlights to us the profound impact on yeast development that ancient, yet inadvertent, artificial selection has had on developing the current yeast strains so widely used in the winemaking industry.
The complete Saccharomyces cerevisiae genome was published in 1996, which, along with improved molecular genetic technologies, has paved the way for the exploration of genetic modification of wine yeast. 2 GM wine yeasts have been approved for commercial use in the US so far. The first is known as ML01 and is a genetically modified strain of Saccharomyces cerevisiae, which has two integrated foreign genes. The first, malate permease gene from Schizosaccharomyces pombe, and the second is a malolactic gene from Enococcus eni. The resulting yeast has both an active transport system that allows malic acid to be transported into the cell, as well as the required enzymes to convert malic acid to lactic acid. As such, malolactic fermentation can occur at the same time as the primary ferment, removing the need for additional lactic acid bacteria to be added and hastening the winemaking process. The second approved yeast is called ECM001, and it's another engineered strain of Saccharomyces cerevisiae, which in this case has an extra copy of the DUR12 gene. This increases the expression of the enzyme urea amidolase, which increases the breakdown of urea that is produced by the wine yeast. This therefore reduces the amount of urea present that could react with ethanol to produce ethyl carbamate, which is a carcinogenic compound. A third target of genetic modification is glycerol production. By increasing the expression of the gene GPD1, the yeast cell's carbon flux can be shifted more towards glycerol production and thus produce less ethanol, a desirable quality for low alcohol wine production.
A current area of research in wine yeast is genetic modification of yeast to produce low or no alcohol wines. As we know, yeast take glucose, and by metabolic processes in the yeast cell, ethanol is produced. Ethanol is not the only byproduct of carbon metabolism in yeast, where there can be acetaldehyde, acetic acid, and glycerol, to name a few. In order to create less ethanol, one of these products will be created in greater amounts. Yeasts are modified by making enzymatic changes in the metabolism of carbon in order to make the process faster or slower, thus producing different amount of byproducts. Traditional PCR, polymerase chain reaction based genetic modification, is time-consuming and laborious. Modern genetic engineering involves clustered regular interspaced short palindromic repeats, or CRISPR technology. CRISPR-Cas9 is much faster than traditional gene editing. First implemented in commercial Saccharomyces cerevisiae in 2013, CRISPR-Cas9 was used to delete gene coding for arginine permease, leading to reduced urea production. Synthetic biology is a larger interdisciplinary field which is aimed at redesigning organisms and engineering them to have new abilities for useful purposes, which is also significantly important on yeast and in the wine industry. The future of the wine industry will be impacted by the outcomes of future holds for synthetic genome engineering.
So, let's talk about some of the cultural implications that arise when we talk about genetically modifying yeast strains for the intentions of creating wine. There are numerous challenges when it comes to genetic modification of yeast strains, such as companies and wineries owning their own strains of yeast, wineries not allowing for the use of genetically modified strains within their wines. On the other hand, however, there are some potential benefits to genetic modification, such as in the creation of lower alcohol wines and enhanced fermentation control. Ethanol production with genetically modified yeast strains has been shown to have been affected overall. The ability to reduce the overall ethanol produced via the process of fermentation has led to the fermented products such as wine and beer having an overall lesser alcohol content, of which impacts the overall characteristics of the product for better or for worse. So, how does this work? This is accomplished by manipulating the gene known as GPD1, which controls the amount of glycerol produced within the process of fermentation. This is arguably one of the most important byproducts of this process, and it serves as a mean to maintain cytosolic redox balance, as is utilized within the osmoregulation of the Saccharomyces cerevisiae species. By increasing the levels of GPD, this shifts the carbon flux towards glycerol, resulting in a lower ethanol production within the fermentation process. Yet another cultural implication is that once a genetically modified strain of yeast is introduced to a winery or a vineyard environment, it tends to stick around and become a part of the system. This can be detrimental to other potential ferments within the winery and inhibit future works. This could, however, be seen as a benefit to specific winemakers and vintners, as having a successful and beneficial yeast as a part of the terroir can lead to potential success within the realms of wild ferments. Finally, some wineries are completely restricting the usage of genetically modified wine yeast within their work, as implications such as risk assessments as well as complete bans on the use of genetically modified organisms are issues that arise. There is a firm reason for this stance to be taken, however. Winemakers are acknowledging the importance of the public acceptance and safety before they adapt and make use of any potential technology within the realms of winemaking, of which should be deeply considered. However, this raises the issue that products could be significantly improved with new yeast strains, as opposed to maintaining what is deemed commercial and successful as of right now.
Traditional selective breeding has evolved many effective yeast strains, but will genetically modified yeasts hold the answer for further improvements for specificity and quality? And will consumers accept it? For that, stay tuned. Thank you for listening to our presentation.